Hi everyone, I'm Sonia Valib.
I'm Eric Minical.
And we'll be speaking to you today about our work to develop an antisense oligonucleotide, or ASO, for the treatment of genetic prion disease. We'd like to tell you a little bit about our story before we tell you about our research. Eric and I are a married couple. We were married in 2009, and in 2010 we went through the process together of losing my mom to a rapid dementia. We didn't know what it was at the time, but she went downhill very fast. She died that year, and afterwards we learned that she died of a genetic prion disease. This told us that I was at risk, and we decided right away to find out if I had the mutation or not. I got predictive genetic testing, and at the end of 2011, we learned that I had inherited the mutation from my mom. So Eric and I were not scientists at the time. I was trained as a lawyer. He was trained as a transportation engineer. But we decided to change careers to try to work on developing a treatment for this disease. We started working in labs, started our PhDs at Harvard Medical School in 2014, and that process brought us to the Broad Institute of MIT and Harvard in Cambridge, Massachusetts, where we're based now, and that's where we're doing the work that I'll be telling you about today. To back up a moment to the genetic testing, before I got my test report, we learned that genetic prion disease is dominant. So that meant I had 50-50 odds of having inherited the mutation from my mom. After we got the testing and learned that I did have the mutation, we learned that genetic prion disease mutations are highly penetrant. So my mutation and a handful of others that cause prion disease give you a very high risk of developing prion disease in your lifetime if you have them. What we can't predict is when that will happen. So age of onset is very variable with these mutations. Basically, disease can strike any time in adulthood. When we were first told about my risk, the term we heard was fatal familial insomnia, or FFI. But we have stopped using that term as well as CJD and GSS and these terms for other genetic prion disease subtypes as our research has advanced, because we've realized that we're really thinking about genetic prion disease as a category. There's one shared molecular mechanism, and all of the therapeutic strategies that we're thinking about would treat them as a class. To say one more thing about our mission and how it's sort of evolved over the years, as we think about where we can do the most good for patients and people at risk, we've really started to focus on the idea of preventing or delaying onset as the place where we have the greatest opportunity. As anybody knows who's been through this disease with a loved one, it moves extremely fast once symptoms strike. And I truly hope that we will be able to do something to help symptomatic patients at some point in the future. But I think that to the extent that we can get to people at risk before they have symptoms, we have the greatest opportunity to preserve and extend healthy life. So you'll hear us return to that theme a few times. So the reason we wanted to speak with you today is that we are optimistic for the first time about a specific strategy to treat prion disease, and that is antisense oligonucleotides, or ASOs. These are drugs that would aim to reduce prion protein in the brain. If all goes well, we're optimistic that an ASO to reduce prion protein in the brain could enter clinical trials within 5 years. So we have a lot to start preparing and start working on to make sure that we have the ability to do the right trials when ASOs are ready.
So we're really lucky in prion disease to know what this disease is at the molecular level, and here's a brief overview. Everyone has the prion protein gene in their DNA. That gene gives rise to the prion protein RNA, which is translated into the normal prion protein, which everyone has in their brain, and most of the time it's not a problem. But it's capable of misfolding into a form that we call a prion, and that misfolded prion can spread across the brain and kill neurons. And from this roadmap, you can see that there's a weakness. You need that normal prion protein in order to have the disease. And so by intervening upstream, we hope that we can delay or prevent that whole process from happening. Antisense oligonucleotides, or ASOs, are a class of drugs that target RNA. So this gives us the ability to target the prion protein RNA in the brain and lower it, thus lowering the amount of the normal prion protein and everything downstream of that. And I want to clarify that even though we're targeting RNA, our goal is simply to reduce the total amount of all prion protein. We're not targeting a specific mutation, and our hope is that one drug will be universal for all different subtypes, all different mutations in genetic prion disease. So how do ASOs work? An ASO is just a short string of chemically modified DNA. It's made of the same 4 building blocks, A, C, G, and T, that make up the DNA in your genes. And the order of those letters, those A's, C's, G's, and T's, is designed to be antisense, or upside down and backwards, to one sequence in one gene in your genome. And so out of all the thousands of RNA molecules in your cells, this gives the ASO the ability to go in and bind to one specific RNA, and it causes that RNA to be cut up. So ASOs are a really precise way to lower the amount of RNA from just one gene and thus lower the amount of one protein. And ASOs are special because the fact that they're made of these same 4 building blocks, just in a different order, means that we already know a lot about how to deliver them to the brain, how they distribute through the brain, their stability and their safety that we've learned from other diseases for which ASO drugs have been developed. And that means these can be developed relatively quickly. In the past few years, there have been some really impressive advances in the treatment of neurological diseases using ASOs. At the end of 2016, FDA approved nusinersen, an ASO drug for spinal muscular atrophy. And it is the first truly disease-modifying therapeutic for any neurodegenerative disease. So spinal muscular atrophy is a disease that in its most severe form kills children by the age of 2. And just a few years ago, it was as incurable and seemingly as hopeless as you may feel prion disease is today. But thanks to that drug, kids who go on the drug, the disease basically stops getting worse. And the preliminary data that have been coming out say that if they're treated before they have symptoms of the disease, those children seem to be developing normally. So it's just an incredibly effective drug and a real story of hope. Another example comes from Huntington's disease, which, like prion disease, is an adult-onset, single-gene neurodegenerative disease of the brain. Some clinical trial data were recently announced, and they showed that they could reduce the disease-causing protein, huntingtin, by 40% in patient cerebrospinal fluid. And remember I said that each ASO is the same building blocks as every other ASO, just arranged in a different sequence to target a different gene. So if it's possible to develop an ASO to reduce huntingtin by 40%, there's no reason in principle why we can't reduce prion protein by 40%. And we believe that if we could do that, it would have a huge impact on prion disease.
So in 2014, we met with the neuroscience team at Ionis Pharmaceuticals, a company that works specifically on antisense therapeutics, including the ones that Eric just spoke to you about. And we sat down with them to talk about how their technology and our disease seemed like a perfect match. They told us that they saw the case, they were interested, but in order to be convinced to work on a clinical drug for this disease, they would need to see some data from us. We left that day with some homework, to do mouse studies to show that an ASO is prolonging survival in treated animals versus untreated animals, to work on a biomarker to show that a drug is having its intended effect in a living patient, and also just to think about a strategy for how we would find enough patients for trials in this rare disease. I'll just show a little bit of the data that's come out of that homework assignment that we've now been working on for a few years. Here is a plot of animals here at the Broad that were treated with ASO and then infected with prions, and then they got one additional dose of ASO, and then we just watched them for a while. And what you can see here is that mice that were not treated fell ill and died of prion disease. Mice that received either one of these 2 active ASOs in blue and green lived about 70% longer. So this was a really powerful and exciting result. And something that I think is even more exciting is that it is now replicated across 2 sites. These are data from our sister site at NIH. The lab of Dr. Byron Coey did a very similar experiment with the same ASOs. And you can see that here too, the treated mice outlived the untreated mice by roughly 70%. This is not a finished story. We have ongoing mouse studies, ongoing biomarker studies, and ongoing recruitment efforts. But I think an exciting update is that we've now gathered enough data on all of these fronts that we were able to go back to Ionis and come to an agreement that we're ready to work on moving this program forward to the clinic. And we're all very excited to do that. We're all committed. It's not a guarantee, and it's very nerve-wracking to say that we hope to be in trials in 5 years, because every day in science brings with it this inherent amount of uncertainty. But we're going to do everything we can to move this to the clinic. So as we think about moving forward to the clinic, now is the time to be thinking about what kind of clinical trials we want to do in prion disease. If you think about a traditional clinical trial, maybe you picture sick patients, some of whom get drug, some of whom get placebo, and then you follow them to see if the patients who got the drug feel better or do better or live longer. That would be the traditional model. This is how most trials are done, and as we said before, it would be great if ASOs can benefit symptomatic patients, and potentially that will be tested. But as we mentioned, our goal is really to prevent or delay onset in healthy genetic prion disease mutation carriers, and that's kind of a different goal. If we're looking to the question of whether a drug can prevent or delay disease, we probably have to test that separately. You can think of, for example, cholesterol-lowering drugs as one example of where prevention and treatment are not necessarily the same. So millions of people take statins like Lipitor to lower their cholesterol in order to reduce their risk of heart attack. But those drugs won't save your life if you walk in the door of an emergency room having a heart attack. And if the bar for getting those drugs approved had been that they had to treat a heart attack the moment it was happening, then they would not have been approved. So as we think about whether a drug can reduce risk, prevent and delay onset, we have to think about doing a trial in pre-symptomatic carriers. The big challenge here is that pre-symptomatic carriers appear totally healthy and have no symptoms. So we can't treat them and ask if they feel better. Instead, what we need here is a biomarker, a laboratory test that tells us whether the drug is having its intended effect on the molecular level. So here's one proposal of what that could look like. The goal of an ASO is to reduce prion protein in the brain. So can we measure the level of prion protein in cerebrospinal fluid? And if that goes down in response to ASO treatment, could that be the basis to get a drug approved? There's actually an FDA program in the US called Accelerated Approval that has enabled this sort of biomarker-based drug approval for other diseases in the past. So could that apply to us? The plan ends up looking something like this. Reduce PRP in the brain with an ASO by targeting the RNA, and then measure PRP in cerebrospinal fluid. In November 2017, we went down to FDA to talk to scientists there about this plan. This was in the form of a critical path innovation meeting, which is kind of an advisory meeting with FDA. It's not a binding meeting, so basically what they can give us is general feedback and ideas. What we heard from them, which was really exciting, is that in principle, it appears that this strategy could make sense. There's still a lot of work to do and a lot of data to gather to assess whether this will be feasible, whether it will make scientific sense. But in principle, we got agreement that it's worth exploring and that trials in healthy carriers could be a path forward. One key assumption that this rests on is that prion protein in CSF is a meaningful biomarker, and it's telling us something about the brain. We have just wrapped up a study where we took a first pass at looking at this and asking key questions about PRP in CSF, how it behaves, where it comes from. And while there's still a lot of work to do in this area, I'm excited to share that this biomarker so far looks strong and like it will support this strategy.
As Sonia said earlier, it's really nerve-wracking for us to put a number up here and say that maybe we could be in human trials in, say, 5 years, because this is science, and things go wrong in science, and things can take much longer than you think. But we realized that what's even scarier is the idea that we could have the drug ready a few years from now, but not be ready because we don't have the biomarker or we don't have the patients. And so we realized that now is actually the time to start getting ready for the right kind of trials, and in particular to start engaging and building the genetic prion disease community. We want people to have the time and the information to make an informed decision about whether they want to participate in research and whether they want to participate in trials down the road. And even more so than any of the things we're doing in the lab with mice and with biomarkers, it was this issue of patient recruitment and engagement that was FDA's biggest message for us. They said this is where a lot of trials can stumble in rare diseases. So one way you can get involved is to go to prionregistry.org. This is a private, secure, online research participant registry. Basically, it's a place you can go, create an account, fill out a questionnaire. It only takes about 5 minutes, and let us know if you're interested in participating in research and trials. So you can find information about studies that are recruiting right now, and you can sign up to be notified when there's a trial in the future. Just by joining this, you are sort of standing up and being counted, giving us a way to show that there are enough people out there that we can do the pre-symptomatic trial that we want to. Meanwhile, here in Boston, we have a clinical research study led by neurologist Dr. Stephen Arnold enrolling at Mass General Hospital. So our biomarker studies to help lay the groundwork for this trial that we're envisioning are relying heavily on samples collected in this study. You don't need to get genetic testing in order to participate. The study involves 2 visits to Boston, 2 to 4 months apart, at each of which you'll get a lumbar puncture or a spinal tap. The study also includes medical questionnaires, cognitive testing, a physical, and a blood draw. And we do have some travel funds available to offset costs. I want to say a little bit more about the lumbar puncture because an ASO drug for prion disease would be delivered by a lumbar puncture. And it's also part of this study. If you enroll in this study, you'll be counseled on the risks of a lumbar puncture before you start. Serious side effects are very uncommon. Sometimes people get a headache. In general, people report that it's much easier than they expected. So here are some quotes from people's experiences undergoing the lumbar puncture in this study. Because this is how an ASO drug would be delivered in the future, we think it's helpful that people are getting these lumbar punctures and can make informed decisions about what it would be like to get a drug delivered this way. Finally, I want to address one issue that it's a bit early to be talking about, but we've gotten this question every time we've spoken about this work. Would I need to get genetic testing in order to enroll in the clinical trial? This is not up to us. This is a conversation between the company developing the drug and the regulators, such as FDA. But from what we've seen in other clinical trials, we guess that the answer is likely to be yes, you would need genetic testing. And the reason is one of risk-benefit. So even though lumbar punctures are quite safe, and even though many animal studies will be done to make sure a drug is safe before it goes into humans, the risk of taking a new experimental drug is never 0. And if you're not sick and you're not at risk because you don't have a mutation, then there's no potential benefit to offset that risk. And so we expect that regulators will say that there needs to be some risk-benefit balance, and therefore only people who've been genetically tested and know that they carry a mutation would be eligible to enroll in the trial. That's one motivation for why we're getting this conversation started now. We want to give people time to make an informed decision, because genetic testing isn't easy, and it's not always a trivial decision you can make overnight.
So that's why we wanted to speak with you today about this project. We're so excited about it. There's a lot of work ahead. Thank you so much to the advisors, allies, and collaborators who've helped us get here and who will be a huge part of moving forward from here. And please stay in touch.